I'll be discussing regarding the biological foundations of obsessive compulsive disorder. Um, Basically, what I'll be doing is I'll be discussing the current understandings and evidence for biological basis of OCD from various perspectives like genetics, neurochemistry, immunology, neuroanatomy, neuropsychology, and neurophysiology. And in the end, I'll conclude with some of the theoretical neurobiological models which have been proposed to explain the symptoms of obsessive compulsive disorder. So now I'll start off with genetics. To begin with, in genetic study, the first step that is usually done is to see whether there's a chance of genetic contribution to the pathogenesis of any disorder. So the first step is aggregation analysis, in which family studies are done to see whether OCD aggregates in families when compared to general population. Since 1930s, a number of studies have been done on this aspect. Earlier studies were with some shortcomings, like they studied only the family history data. They didn't do direct interview of the relatives, and some studies didn't include controls also. But later studies, especially after 1987 or 90, were done more methodologically rigorous, and a larger sample size were used. And as a summary, most of these studies have shown a higher prevalence of OCD in first-degree relatives of patients when compared to age and sex-matched controls. Different studies have shown different prevalence rates for relatives. A meta-analysis in 2001 has shown that OCD, the relatives have 8.2% prevalence compared to 2% in controls, which is almost equivalent to the prevalence in the general population. So, to summarize, it can be said that first-degree relatives of OCD patients are 4 times higher the risk of developing OCD. But when the studies were categorized into adults as proband studies versus children as proband studies, it was seen that the children with proband studies have more consistently demonstrated higher prevalence of OCD. And it was also shown that the exact prevalence was higher in relatives of children with OCD when compared to adults with OCD. So it can be said that children with OCD have a higher genetic contribution to the pathogenesis of OCD. Apart from OCD, it has been seen that in relatives, other disorders are also very common, like other OC spectrum disorders. They have around 2.7 chances of increased risk of having OC spectrum disorders. And some studies have shown that in the same way, like early onset OCD has a higher comorbidity of tic disorder, and late onset OCD has a higher comorbidity of anxiety disorders. So there is a possibility the different genetic contributions contribute to early onset versus late onset OCD. Coming to the next step, that is segregation analysis. Although it has been seen that OCD aggregates in families, this aggregation can be due to either a common genetic contribution or a common environmental contribution. To confirm that, twin studies have been done, and many twin studies have been done with large sample sizes. And many studies have seen for obsessive compulsive symptoms in twins. They are not seen for disorder, but still what they just wanted to see is whether there's a propensity for obsessive compulsive symptoms in twins. What they found is, for example, in the study, which includes 854 twin pairs, monozygotic twins had 57% concordance in OCD compared to dizygotic symptoms of 22%. So since the environmental influences both in monozygotic and dizygotic, the rest 30% can be attributed to genetic factors. To see how much a percentage of this concordance is due to genetic factors, various studies have been done, and the results vary. But similar to those in family study, it has been seen that heritability, that is the measure of genetic contribution, is higher in children when compared to adults. Similar results have been seen not only for symptoms, for syndromal OCD also. And these studies have also shown that the genetic factors of OCD is also shared. That is, the twins are concordant for tic disorder, and some studies have shown that twins are also concordant for anxiety disorders. This study by Tams et al., they tried to find the genetic heritability of OCD, and they found that 55% of the genetic factors are common to all anxiety disorders, and the rest 45% is only specific to OCD. This goes against the evidence that people whether to separate OCD from anxiety disorders or whether they share a common pathogenesis. Apart from that, people also have tried to do a complex aggregation analysis. Basically what it does is we find the mode of inheritance. Based on statistical modeling, we try to find the mode of inheritance. It has yielded inconsistent result, but there are few studies which have shown that there may be a single or few genes which show major effect, especially when they study a particular part of the OCD sample. For example, they have seen that hoarding sample may be, patients with hoarding may be contributed by a single gene, and females may be contributed by a single gene. But these are inconsistent results and needs to be replicated. And there is also an evidence that the heritability is different for different symptom dimensions. Now coming to find, so although it has been established that genetic factors contribute to OCD, what are the actual genes? So initially started with the linkage analysis, where we find to see which part of the chromosome is responsible for transmission of OCD. Multiple different studies have yielded different parts of chromosome. There is only one chromosome, that is chromosome 9p24, which encodes a gene for, it's nearer to the gene which encodes glutamate transporter. This chromosome has been replicated twice, but all other chromosomes have not been adequately replicated. This is followed by candidate gene studies, where association genes studies are done. Genes encoding for a specific function is compared between patients and controlled. People have tried to study genes related to the commonly hypothesized neurotransmitters like serotonin system, dopamine system, and all other systems. And here also there have been results, but replication is not adequate. Except similar to the linkage studies, the glutamate transporter SLC1A1 gene in chromosome 9p24, this has been replicated in 4 studies. So to summarize the genetic studies, it can be said that there's adequate evidence that OCD has a genetic basis, and people have hypothesized that it's an oligogenic disorder with complex gene-environmental interaction. And different subtypes of OCD may have different genetic contributions. Like childhood OCD may have a different genetic contribution compared to late onset. And similarly, patients with tic disorder also may have a different genetic contribution, and similar to symptom dimensions. And regarding specific genes, the only gene that has been replicated is the glutamate transporter gene. So coming to the neurochemistry, various neurotransmitters have been studied. I'll be discussing only the common ones, mainly serotonin, dopamine, opioids, and glutamate. In serotonin, as we all know, the serotonin hypothesis of OCD started because of better response of OCD to serotonergic reuptake inhibitors compared to other antidepressants. Although this evidence was there, people have studied whether there is any serotonin pathology in OCD patients. Some measures, like the indirect measures, were made, like peripheral receptor binding in blood cells, platelet serotonin binding, CSF metabolites of serotonin. All these studies have revealed inconsistent results. Some challenge studies were done, like this MCPP, which increases serotonin level, and this also has shown positive results in 4 out of 6 studies. There were 2 negative studies. When it comes to direct measurements in the brain, it has been found that using PET and SPECT studies, serotonin transporter binding in OCD patients have shown inconsistent results. Gene study, this gene 5-HTTR promoter gene, this has been famous because of Caspi et al. Study, which has shown to be associated with many neurotic disorders, and especially depression. An interesting finding in OCD is regarding OCD is that most of the studies in neurotic and depressive disorders have focused on the S allele, and they have said that S alleles, presence of S allele is associated with risk for depression or neurotic disorder. But in OCD, it is inconsistent, but the studies which are shown show more contribution of L allele rather than S allele. So, although we don't have evidence to suggest that serotonin doesn't, it's not directly related to the pathogenesis of OCD, how do SSRIs act? It has been seen from animal studies that when SSRIs are given, the receptor changes occur more late in the orbitofrontal cortex compared to the prefrontal cortex. So this is somewhat we can see an analogy between response in depression and OCD. So maybe the late onset changes orbitofrontal cortex maybe may cause the decrease in OC symptoms. So it has been hypothesized that downregulation of IHT1D autoreceptors in orbitofrontal cortex decreases obsessive compulsive symptoms. But since it is not directly implicated in pathogenesis, it can be hypothesized that the serotonergic system is overcompensating for imbalance in some other system. The second most common neurotransmitter that has been studied is dopamine. Dopamine in animal models using transgenic mice, which increases dopamine transmission, and also some pharmacological challenge studies with dopamine agonists. These studies have shown that when increase in dopamine is there, the animal starts expressing repetitive behavior, which is similar to OCD, something like excessive grooming. But the problem is that these may or may not be considered as compulsions. We don't have any direct evidence for obsession, but even if it takes us at a compulsion, it's difficult to differentiate from stereotypies. There's also human evidence, like certain dopamine agonistic drugs may induce OC symptoms in humans. Regarding genetic studies with related to dopamine genes, the results are inconsistent, but there is a hint that in patients with OCD, presence of a certain alleles of dopamine genes related to dopamine receptors is associated with the presence of tic disorder. So OCD with tic disorder may have some pathogenesis related to dopamine. Now going to the direct observation of dopamine in the brain, it has been found that dopamine receptor in the anterior cingulate shows a decreased binding, but in basal ganglia it has been inconsistent. Treatment response literature shows that dopamine antagonists may be effective in certain subsets of patients, like people with comorbid tic disorder or schizotypal disorder. So to summarize, it can be said that dopamine doesn't have any direct effect on most patients of OCD. There is a possibility from the current literature that a subset of OCD population, like those with tic disorder, may have some pathogenesis related to dopamine. Next is regarding opioids. As we know, opioids have been used in certain impulse control disorders. The pathogenesis of opioids in OCD started when people found that people with OCD who are comorbid with opioid dependence, when they were OST, when the dose of methadone was tapered, the OCD symptoms started worsening. So people felt that maybe opioids are beneficial in OCD. And there is some evidence to show that in people with OCD, there's decreased endogenous opioid levels. So some treatment studies have been done. These studies have shown that certain opioid agonists are effective as an augmenting strategy in treatment-resistant OCD. But these studies needs further replication. Mechanism I'll be discussing in the further slides. Now coming to the neurochemistry of glutamate. Currently this is one of the hot topics in neurotransmitter research in OCD. The CSTC circuit, which is implicated in OCD, it has been found that through studies that glutamate is the primary neurotransmitter in that circuit. And even in striatum also, the majority of axon terminals are glutaminergic. So there's a possibility of glutaminergic thing in OCD. And animal models, similar to in dopamine, it has been found that certain animal models which have either to transgenic or removing certain genes are found that any increase in increase in glutamate levels is associated with certain repetitive behavior. As summarized in a previous genetic study, it has been found that this is the most commonly reported gene. That SLC1A1 is the most commonly reported gene in replicated gene in OCD. So direct measurements, CS of glutamate has been found to be increased in patients when compared to controls. So there's a possibility of glutamate pathology. And direct measurements using MRS technology has found that glutamate transmission, glutamate concentrations are increased in the left but not the right caudate nucleus, orbitofrontal, and low in the anterior cingulate gyrus. The interesting thing is that all these things have been implicated in the pathogenesis of OCD. And there are some immunological studies which also give an indirect evidence of glutamate pathology in OCD. For example, one study have found that this circulating immune complexes specific to Borna viruses have been shown to be associated with OCD. And there is another study which has shown that circulating immune complexes actually increase glutamate level. So, keeping all this in mind, there is some evidence which suggests that glutamate has an important role in the pathogenesis of OCD. Coming to the third topic, the role of immunological factors in OCD. As we all know, the most famous among this is PANDAS. This pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections. It was hypothesized that certain people with rheumatic fever develop neuropsychiatric symptoms, especially obsessive compulsive and other obsessive compulsive spectrum symptoms like tic disorders. And these symptoms occur along with other motor manifestations like sedentary chorea. And they hypothesized that maybe these symptoms are also due to autoantibodies, similar to sedentary chorea. And there are some evidence for this thing, like the OCD symptoms in PANDAS patients have an episodic course, and the symptoms are closely associated with motor manifestations. And when MRI studies were done, it was found that during the symptom period, there has been enlargement of basal ganglia, and after the patient went into remission, the enlargement decreased. Another important factor is that some immune modulating treatments like plasmapheresis have shown to have some response to in PANDAS. But the direct evidence finding of autoantibodies against basal ganglia has not yet been consistently replicated. So this still remains a hypothesis. Apart from streptococcal infection, recently there has been hype about Borna virus and neuropsychiatric syndromes. It has been studied in most of psychiatric disorders, and similarly it has been done in OCD also. But it still remains to be confirmed whether this is just a fad or this is, has any evidence. In general patients with OCD also, a few researchers have found there is autoantibodies against basal ganglia and thalamus. So autoantibody research have shown inconsistent results. So other peripheral inflammatory markers like cytokines have been studied, which also have shown inconsistent results. There is some age relation. It has been shown that younger patients have higher disturbance in cytokine levels. So this also may point out that younger onset OCD may be more related to autoimmune pathology. But this is still hypothesis, needs more replication to find out the final. The next type of study is neurophysiological study. In this study, I will just be focusing on this particular aspect, intracortical inhibition. Intracortical inhibition is measured using transcranial magnetic stimulation. What is done is magnetic stimulus is provided on the motor area of the brain, and when the magnetic stimulation is given above a certain level, which is called the threshold level, motor evoked potential is seen in the corresponding area. Usually we try to see in the thumb, the abductor pollicis brevis. When this is done, it can be measured by EMG. And in normal people also, what has been seen is that before the magnetic stimulation is given, a subthreshold stimulation, not exactly in the same area, somewhere around the area, if it is given, the amplitude of the EMG response decreases. Okay. So people have hypothesized that this seems to be due to some GABA-dependent interneurons, which decreases the firing of the certain neurons. And they have found that this is present in almost all normal people, but in OCD it has been seen that this intracortical inhibition by a previous subthreshold stimulus is decreased. So people have hypothesized that there is a deficient functioning of the interneurons, GABAergic interneurons in OCD, and this may lead to OCD. What they have hypothesized is that these GABAergic interneurons, they decrease the firing of unwanted impulses in the brain. So when this is not there, unwanted thoughts come repeated to the consciousness. But this is just based on one study. Further studies are needed. Neuropsychological studies on OCD also have shown some interesting data. The most commonly reported neuropsychiatric deficits are response inhibition, set shifting, decision making, and nonverbal memory. All these have some relation phenomenologically with OC symptoms. There is one aspect. The other aspect is that all these functions are related to the prefrontal lobe function, which is also implicated in OCD. And these neuropsychological deficits are also seen in relatives, unaffected relatives of OCD, and also when the patient is in remission. So this is a possible endophenotype marker of OCD. But different studies have found different neuropsychological deficits, none of which it is common to most studies. Now coming to the neuroanatomy of OCD, I'll be focusing on the structural imaging and the functional imaging part of OCD. Chemical imaging has been discussed in the neurotransmitter section. Structural imaging findings have found the same circuit, left anterior cingulate and left and right orbitofrontal cortex. They have been found to have reduced volume. And it is also found that there is increased volume of the thalamus. And regarding caudate, it is inconsistent. This is a review of studies based on region of interest based MRI studies, and it can be seen that orbitofrontal cortex has more replicated studies, and similarly thalamus. Caudate is plus minus. But one disadvantage of ROI based MRI studies is that it is able to study only large structures. To study smaller structure, a more sophisticated methodology is necessary. That's why maybe caudate may not be able to pick up in those studies.
Dr. Sundar, would you please summarize in a 5 minute?
Okay, I'll summarize.
5 minutes. Okay.
I think this has shown that caudate may be involved. Functional imaging also has shown similar results, and symptom provocation also have shown similar results. Based on this is the model that has been proposed. A circuit from cortex, striatum, thalum, basal ganglia to the thalamus. This is a closed loop which has been implicated in OCD. Many such loops exist. Each loop is associated with different symptoms, and it has been hypothesized that each loop is associated with different OC spectrum disorders like tics, anxiety, OC symptoms, and attention deficit. And certain symptom provocation studies have also lent support to this model. I think this will be okay. This is also another hypothesis called implicit process and deficit hypothesis, which has been proposed to explain OC symptoms. So to summarize, it has been seen that genetic factors contribute to at least certain types of OCD. CST circuit seems to be involved. Although serotonergic agents are helpful in management, there is inadequate evidence to show that they are involved in pathogenesis. Currently, more evidence suggests towards glutaminergic system, but intervention studies have to give the final say. Thank you.